Hi there, and welcome back to ESMARConf. We have a really interesting session for you now, focusing on research synthesis. First up, we have Sarah Young, and Sarah Young is going to be talking to us about what networks can do to help support evidence in evidence maps. Over to you to share your screen, Sarah.
Okay. All right. Hello, everyone. Happy to be here and presenting to you about this very new project, looking at how network analysis can be applied in an evidence and gap map context. So evidence and gap maps are, whoops, sorry about this, technical issues. There we go. Evidence and gap maps are an evidence synthesis method that asks somewhat broad questions about what knowledge exists related to practice and policy relevant topics. They also highlight knowledge gaps and knowledge clusters and can be used to guide research priorities moving forward. So they typically include an analysis of kind of a thematic sort of summary of the evidence. For example, you can see here a thematic matrix that is sort of an interactive tool that policymakers and decision makers can use to understand what evidence exists. They also often do analyses over space and time. So for example, they might look at the geographic location of studies or the numbers of publications over time. However, the work of Nakagawa and colleagues in 2019, which introduced this research weaving concept, really highlights the fact that there are some real missed opportunities here with regard to what more these evidence and gap map data can tell us. And so the purpose of this project is really to demonstrate what these methods can add to an evidence and gap map. And particularly today, I'm going to focus on network analysis methods. So network analysis essentially adds a new dimension of study to the evidence and gap map, namely a social dimension in which we can sort of begin to understand how this body of knowledge is structured through collaboration and citation behavior. And this could ultimately inform recommendations for future research, as well as aid in the discovery of additional relevant literature and terminology. So several open source tools exist to facilitate this kind of analysis. Gephi and VOSViewer are two popular tools, but given the focus of this conference, I'm going to really focus on R. And in R, I'm going to highlight the Bibliometrics package, which offers a suite of functions to carry out many different types of network analysis using bibliographic data. And it typically works with standard bibliographic databases like Scopus and Web of Science. And you can use functions in the Lit Searcher package, if you're familiar with that, to build an exact title search from your list of studies in your map to then run in a database and then export a file that can be analyzed in Bibliometrics. So that's sort of a nice little workflow that can be implemented in R. So there's a lot of different types of networks that can tell us different things about this research community. So for example, relationships between researchers based on shared authorship is represented in a co-authorship network. And this type of network can really help us identify key researchers or identify research silos and can indicate something about the flow of knowledge and information across the network. So this image here was generated in Gephi. There are also ways we can look at relationships between studies based on cited references. And these are usually conveyed as a co-citation network or a bibliographic coupling network. And this visualization was generated with Bibliometrics in R. And it's an example of a co-citation network where you have the nodes represented as studies and relationships between those nodes when they are cited by the same study. And finally, we can also look at bibliographic networks based on the co-occurrence of terminology. This is a keyword co-occurrence network, and this can also indicate topic-based communities and allow for further analysis of collaboration and citation behavior based on topics. And they can also show us new terminology that can be used to improve or update an existing evidence and gap map. So for the purposes of this project, we're going to take sort of a case study of a previously published evidence and gap map. In this case, one on the multifunctional roles of vegetated strips. And the idea here is to sort of demonstrate what these techniques can bring to an evidence and gap map and provide tools, again, using R and other open source tools that can be applied widely across evidence and gap maps across domains. So this particular evidence and gap map, in addition to sort of the standard analyses that are done, did some really interesting text analysis and found just a remarkable array of terminology that's used to describe vegetated strips. And so I think this is something we can probe a bit more using a network approach. There's a bunch of different questions we could potentially ask in this project, and these are just some initial ideas. But ultimately, we're trying to understand, what is the structure of this research community? What is driving that structure? And ultimately informing, what future research should look like and how this kind of information can inform that. And also thinking about how this technique can help us find new terminology and discover new research. So I do want to mention just a couple of considerations here and challenges. For example, with co-authorship networks, it's very important that author names be disambiguated appropriately and accurately. This can be quite onerous in a large data set, but tools like OpenRefine can help here. And there may be tools in R, and we certainly welcome suggestions for R packages that might assist in this kind of data cleaning. Additionally, I mentioned Bibliometrics relies on outputs from Web of Science and Scopus. And of course, in an evidence and gap map, all of that literature may not be indexed in those databases. So there may be some significant amount of work getting all of your bibliographic data represented in a file that works in this package. So I'll go ahead and wrap up there, but this is a project that also involves, I should say, Neal Haddaway and Shinichi Nakagawa. I believe Max Callahan will be joining us in the very early stages, and looking forward to any thoughts or suggestions that anyone has.
Great, thank you so much, Sarah. That was really interesting. Obviously really interesting, but me because I'm involved. But I hope it was really interesting for everyone else as well. That was a great talk. So next up we have Tim and Dorian, who are going to be talking about analysing Canadian ecological restoration literature using bibliometrics and systematic mapping.
All right. Hello, everyone. Thank you for joining us for a brief presentation on a project that me and Tim have been working on for the past 6 or 7 months. And it's called Analyzing Canadian Ecological Restoration Literature with Bibliometric Analysis and Systematic Mapping. In this presentation, we will first discuss our overall approach, followed by our use of Biblioshinee, which is a tool in the R environment through Bibliometrics that is used for conducting bibliometric analyses, and then how we took that data, refined it into a systematic mapping approach using programs such as Schema, Tidyverse, QGIS, to extract meaningful information about the trends in Canadian ecological restoration literature and opportunities for the future.
So this project was funded by the Tri-Council Agencies, which is a national funding body here in Canada. And they have a pool of money that's set aside specifically for evidence synthesis, which is pretty cool. This is a collaboration between UVic, Laval, University of Ottawa, and University of Waterloo. So we had a pretty big team. Dorian and I were given a very broad mandate. We were essentially told to synthesize the Canadian literature on restoration and go ahead. So our first task, our first question was, what is restoration? We decided to take a pretty broad approach. You can see from the first half of our search string here, we're including terms like reclamation, rehabilitation, reforestation to try to capture the whole scope of restoration practice. The second question we had to tackle was, what is Canadian? We decided for the bibliometric analysis piece of our research to include any study with an author with a Canadian affiliation or that was located in Canada. We limited that a little bit more for the systematic map piece. And then we had to answer, what is research and how do we include the gray literature in this? So here's a sample search string that we used for our bibliometric analysis. It captures kind of the broad sweep of restoration and Canadian broadly construed. We approached the project in two phases following Nakagawa et al.'s research or protocol here. We were looking at a synthesis of both evidence and influence to try to answer what is published and what is studied. We divided this into two phases to make it manageable. It ended up being that the research question shifted slightly for the second phase because we had to limit the amount of studies we were looking at. The bibliometric analysis included some 3,000 studies. There's no way we could extract data from that many. So we had to limit it to research that focused on the outcomes of ecological interventions. I'll pass it over to Dorian to talk a little bit about the bibliometric piece of things.
Yeah, so our bibliometric analysis was performed in the Biblioshinee interface environment. And the interface itself was really easy to use. It's really clean and intuitive. It's mostly a point and click interface, so there's no coding by the user required. It allowed us to import a large data set of literature from Scopus because it's compatible with the program. And that included standardized metadata about author, year, institution affiliation, keywords, and citations. Biblioshinee offered many options for the actual analysis itself, like Sarah brought up in the previous talk. And they explored the structure of knowledge that Biblioshinee kind of groups into three main categories. So the first is conceptual, which they refer to as the relationships between themes and keywords. So here's an example of one of the co-occurrence network analysis that was outputted for us, wherein the centrality and the size of the terms and their bubbles represents their frequency and prevalence within the literature data set. The width of the connections between terms represents the strength of the connections, and the colors are program generated to represent different thematic bodies. Options for layout, normalization, and clustering in all these figures are available to the user and are easy to experiment with. We also had the option to work with thematic evolution maps, and that allowed us to divide the themes into time slices and see the evolution of themes over time. The second structure of knowledge is the intellectual one, and that refers to the relationships between the papers and the intellectual output. So here's an example of a co-citation network analysis, which basically looks at the connections between citations and references to differentiate group themes. We also have historical direct citation graphs, which take the network analysis and sort of plot it as a function of time to see the evolution of those themes over time. And finally, we had the social knowledge structure, which referred to the relationships between authors, institutions, and countries. So shown here is the collaboration between authors and institutions in a network map in terms of their association with countries. So there's strong connections between Canada, the US, and China. And then we can also project that as a world map, wherein the width of the lines represents the strength of the connections. Some of the challenges with using Biblioshinee for these figures included the screening portion, which there wasn't really an option for that in the Biblioshinee environment or in the Scopus environment. So we had to take an extra step, upload it to Excel, and screen from there, and then re-upload it to Biblioshinee. Figure quality was a little bit of an issue when keywords or citations within a crowded network web were small, fuzzy, and difficult to read. And on that note, the options for the graphical personalization were pretty limited, making it difficult to customize outputs in useful ways.
And for the systematic map piece, we kind of went back to the beginning and revised our search string to focus on studies that document the outcome of ecological interventions. This time we were interested only in studies that occurred in Canada, regardless of where the author's affiliation was. We used Kadima to keep track of the search results. It's a free online web-based platform. It is great for effective and efficient screening. It keeps track of every exclusion that you make. It also presents this form for data extraction, which was great and very helpful, but had one significant drawback. And that is that you could not select multiple categories when you're selecting categorical data. So for example, under research province, we had a lot of studies that occurred in Ontario or in Quebec and New Brunswick. We couldn't select both of those. So we got around that by using the semicolon as a separator, but that kind of messed up our data set and meant that we had to use tidyverse to parse it out. So we wrote a R function that would clean up the data and parse out those columns and then produce heat maps and bar charts. We also use QGIS to do a geographical map of it. Of course, every study uses its own format for the coordinates, but they were relatively easy to transpose and make the same. So I'll show some examples of our systematic map graphics. We use ggplot in R to make these heat maps. It works pretty well. Thanks to a custom function, we could make a heat map for any two columns. That allowed us to kind of play and explore the data really, really easily. Just by writing, a single line of code, we could get a new heat map and see, does this tell us anything about the data? We're starting to learn a few things about the concentration of ecosystems. For example, it's very interesting that Prince Edward Island, which is an island, doesn't have any marine or coastal restoration projects on it being studied. We also looked at ecosystem by publication year, which shows some time-based trends. Bar charts were very simple, but very revealing. It's very interesting that all of the restoration studies are really concentrated in 4 provinces: Alberta, Ontario, Quebec, and British Columbia. There is a lot of ecosystem degradation in the Yukon, Nunavut, and Northwest Territories in the form of mining exploration, but not very many on-the-ground restoration studies.
So bringing it all together, our objective in this project was to take a database of literature that's quite extensive and hasn't really been synthesized before, and make it useful for not only academics, but practitioners as well, by allowing them to easily identify trends, strategies, and opportunities in ecological restoration. Biblioshinee provided the groundwork, highlighting trends, relationships, and connections between individuals, ideas, and locations. Systematic mapping allowed us to extract the finer scale information about ecosystems, methodologies, and outcomes. Our team will be developing a comprehensive report using not only our literature-derived results, but also results and themes from interviews with practitioners and case studies from across Canada. So we're hoping that kind of synthesis from a broad variety of viewpoints and data sources will reveal some interesting information about the direction of ecological restoration in Canada. So thank you all for listening and taking part today, and we look forward to any questions from the audience.
Thanks so much.
Thanks so much, Tim. Thanks so much, Dorian. Really interesting discussion there of combining systematic mapping and bibliometrics using packages I definitely wasn't familiar with before. So thank you very much. Next up, we have Lottie Gasparini, and she will be talking about metalabR.
Welcome to Introducing metalabR, a package to facilitate living meta-analyses and dynamic meta-analytic visualizations, which I'm presenting on behalf of the metalabR Consortium. At metalabR, we are interested in language and cognitive development in infancy and childhood. In early language development research, the overarching question we have is how and when do babies learn language? But language is an incredibly complex system made up of many aspects, like sounds, words, and grammar. So to answer these questions, we need to break it down by asking questions about the various steps along the way that lead to learning language. Questions such as, when can babies tell the difference between 2 different languages? Or between different vowels, like I and U? When do babies figure out when one word ends and the next begins? When do they start recognizing words? And when can they recognize or learn words that are embedded in a sentence? So when conducting studies and reading the literature, we're often looking for results that tell us babies do X at age Y. But such statements made on the basis of the results of a single study can be problematic. Significant results could be a false positive, or null results could be a false negative, especially in infant research, which is notoriously underpowered due to the difficulties of recruiting and testing babies. How do we know if a study's results can be generalized to all babies? They might actually be restricted only to babies learning a particular language. Or perhaps the results only apply in a specific lab setting or when using particular stimuli and methods. Systematic reviews and meta-analyses help bring coherence to a complex evidence base and improve our confidence in results. But meta-analyses are underused in developmental research. To assist developmental researchers in conducting and accessing meta-analyses, my colleagues created metalabR, a platform for open dynamic meta-analytic datasets. This goes beyond a traditional meta-analysis to what we call community-augmented meta-analyses, or CAMAs, which allow for the smooth addition of new data points to ensure that quantitative syntheses give the most up-to-date summaries of the body of literature. In 5 years, the site has grown to 29 meta-analyses with data from almost 45,000 infants and children. And now on to the exciting part for this presentation, our metalabR package, which allows us to take full advantage of the large array of the metalabR data by conducting quantitative synthesis in R. The 3 overarching package features I'll be presenting in this presentation are functions for reading live metalabR data in R, compatibility with running meta-analytic models using Fichtbauer's metaphor package, and visualization tools. So first, the processes of reading the data. To give you a sense of the data curation process, the user conducting a meta-analysis extracts data from studies into this metalabR spreadsheet, which has standardized column names regarding methodological details, participant details, and the quantitative results for calculating effect sizes. Back in R, the getMetalabData function in the metalabR package reads the specified dataset live from the Google Sheet. If needed, the function can synthesize multiple datasets on different topics. Behind the scenes, the data is validated and cleaned to ensure adherence to the standardized data format. Then the function computes standardized effect sizes from the values provided in the Google Sheet. And finally, the function returns a tidy data frame. Fichtbauer's metaphor package already has many great functions for running meta-analytic models, so the metalabR package has been designed so that the resulting data frame from reading the data is compatible with Fichtbauer's metaphor package, running various analyses like calculating estimates of effect sizes and assessing the significance of moderators. And finally, the metalabR package contains various functions for visualizing effect sizes, such as a forest plot, violin plot, and funnel plot. These build on the ggplot package, but provide by default an APA theme and selected colors, as you can see here in these plots. This makes the process of plotting quick and easy and gives the metalabR plots a standardized look and feel. As well as using the metalabR package in R, the package is the basis for the Shiny apps that are up on the metalabR website. These applications are visualizations for exploring the results of existing datasets, power analysis and power simulation tools to help researchers plan their own studies, and data validation tools to assist reviewers to make sure their planned meta-analysis fits the metalabR structure and format so that their data can eventually be integrated into metalabR. And this screenshot gives you a sense of the easy user interface of the visualization tool, where you simply select a dataset from the drop-down menu and visualizations are automatically generated. So in this presentation, we've seen the functions of metalab and metalabR that facilitate conducting a meta-analysis on a new topic, that help researchers plan a new study on a topic where a meta-analysis has been done, help them to add new data to an existing data analysis, and to conduct a synthesis of data across multiple topics of research. These synthesis tools can help us to move beyond the results of single studies and studies researching a single level of language to integrate findings from different areas of research so that we can get closer to answering the question that we're all pursuing: how and when do babies learn language? Thank you to the MetaLab team, as well as the authors and participants of original studies and meta-analyses. Thank you.
Thanks so much, Lottie. That was a really interesting talk. So it's really fascinating to see the different approaches to, I guess, the concept of weaving different things together, whether it's weaving different disciplines or different aspects of the same question we saw in Lottie's talk, or whether it's weaving together different schools, the science metrics, bibliometrics, and the evidence mapping approaches. We're incredibly lucky to have Shinichi Nakagawa here with us, who coined the term research weaving. So I'd like to invite Shinichi and the panelists to come back and just have a chat about some of these approaches. I've got some questions, but Shinichi, do you have any immediate comments? We can all turn our videos on if you want as well. Lovely to see you.
Hello, yeah, thank you. Yeah, it's been really interesting talks, and it's great to see that people are thinking about in different ways to using, especially how to use bibliometrics to match the, how to incorporate into systematic reviews, and also meta-analysis stuff, lots of visualization. I wonder the one thing is that, Sarah talks about bringing social dimension. And last talk, I was thinking you have a lots of really nice visualization. This metalab, Shiny app or a framework looks really, really fantastic. One of the things we discussed in our original paper, Neal was part of it, was independence of evidence. So what we can do from bibliometric analysis is, meta-analysis, we are treating all those, each effect size independent, but really, how many groups are there in this particular topic? Maybe if you have a 10 independent group, it's more reliable. If only 2 groups or 1 big group, how reliable it is, and what is the influence of particular group. So that's something we haven't talked about, but that's something bibliometrics analysis can offer to meta-analysis and systematic reviews.
Yeah, that's a really good point. Does anybody want to respond to that point by Shinichi? I had a sort of related question, I guess. With each of these types of analyses, whether you're doing a bibliometric analysis or an evidence mapping analysis, the analyses and the visualizations are very complex. As we saw in one of the earlier sessions today in DataViz, visualizing evidence bases are very complex. And I was wondering what people feel about the need for specialist communication platforms to combine those different types of data and the different types of visualizations. How do we go about combining a complex network in concepts and a complex network in people? So it follows on from your point there. I don't know if you have any thoughts on the tools that you've used so far, Tim, Dorian, Sarah, and Lottie, and whether you have any concepts, any thoughts, Shinichi, in terms of the concept of research weaving and platforms for communication.
I would say from our perspective, our research was a little challenging because we kind of have 2 audiences. Ecological restoration is a practice, and restoration ecology is the science around that practice. And we are trying to cater to both of them. So while we're producing our report, we're thinking about, what do academics care about? What does the restoration ecology side care about? And what do the practitioners care about? And they're different things and they're different buckets. And so one of the things we really struggled with is figuring, how do these network graphics and heat maps and so forth, how do they translate to the practitioner side? And what can we provide to the practitioner side? Dorian gave a little glimpse of this at the end, but one of the things that our database did is it described these 300 papers. So we're possibly looking at making that into like a WordPress table that you can easily sort by like ecosystem type, so that practitioners, if they're interested in, say, wetland restoration, could look up, all the papers on wetland restoration in our data set. But yeah, one of the challenges is certainly having a surplus of graphics and having to pick, what tells us meaningful things about this data set.
Yeah, and I would just add, we're in the very, very early stages of our project, and I think just from doing some initial analyses, it seems very easy to head down any number of rabbit holes with this kind of work. I mean, there's so many things that you could explore here. And so, we're talking about, well, maybe we should sort of revisit the authors of the original paper and really talk to the stakeholders in this project, and think about what questions do they really want to ask, that might be answered by a network approach. And so sort of using more of a question-driven approach instead of just, a free-for-all of analyses.
Great, thank you so much. We have one specific question that's come in from Elena Ticola to Tim and Dorian. Thanks very much for the talk. In which way is a big number of studies a problem? Was it an issue computationally, or that it didn't make sense to use so many studies to answer your research question?
Yeah, there definitely are limitations in terms of how many papers you can upload to Biblioshinee at once, if I recall, through Scopus, or the other option is Web of Science, I believe. But the program was able to handle that volume of metadata, no problem. Screening 2,000 plus papers is laborious, but it does help that things are becoming more standardized, so that you don't have to filter through a mess of data. You just have everything in columns for you ready to go. Tim, do you have any additional?
Yeah, the big challenge was the systematic map piece, because all of that data had to be manually extracted. So, in terms of like what ecosystem the study focused on, what province it took place in. So Dorian and I managed to do 300 papers ourselves, and it was just the two of us on a research assistant budget doing this work. And so that's how we had to take a really hard look and say, what's realistic for us to do a systematic map of, and we were fortunate that, by narrowing the question to the outcomes of ecological interventions within Canada, we were able to get a manageable number of papers. If it was unmanageable, we may have considered date limiting it, like eliminating some of the older results to make it more manageable. But like Dorian mentioned, the screening of results took a long time, and the extraction of the data it just takes a long time.
That's a really good point. The systematic mapper in me wants to know which is the most painstaking and painful part, the systematic mapping or the bibliometrics. I think I know the answer.
Definitely the systematic mapping part. That's tricky. And one of the big tricks that comes up is you think you have a good number of fields defined, and then you start going through the studies and you get to, study 50, and you're coming up with new ways that people are studying things, and your fields no longer seem relevant. So, it can be really trying that way. One of the innovations that we came up with to kind of deal with that, Dorian, maybe you can talk about the aggregate columns, like that secondary analysis that we did.
Sure, yeah. In expectation of something like that happening, where further down the line we would realize that additional details might be relevant. We took columns that were initially more specific data we extracted, and then once the extraction was complete, we go back, we sort of aggregate those columns into more course level categories, so that the information doesn't have something like 30 to 50 different terms involved with it. It's hard doing a heat map, for instance, with that many terms for each axis. So aggregating them more coarsely might actually be easier to visualize. The information is easier to communicate, and it could actually give some more useful direction to anyone who's looking at this data and this information. I will give an honorable mention to the initial screening process, which was upwards of 3,000 papers with probably the widest variety of topics I could come up with. So there's the terminology you have to go through to sort of understand what they're talking about and fit it into whether it's relevant to your question or not is difficult. At least further down the line, you have sort of the language sort of gets more standardized. So your brain isn't going from one topic to doing a 180 to a different one.
Thanks very much. That's a really good point. One piece of advice I would make perhaps is that if you're using a tool that doesn't screen using RIS files, you have to think quite carefully about how, if you're doing the systematic mapping first and you end up with an Excel spreadsheet where you just have the author and the title, how are you going to link that back to rich data on author networks? Because if you summarize the author's surname and first name, it might not be in the same format that the original RIS file is. I know that from experience because I did exactly that with the original systematic map that Sarah's had to deal with. But I think that if there are tools that use RIS, that makes it much easier to then perform the network analysis and the result, I guess. There's a question for Lottie. Emily Hennessy asks, in case there are other teams who are interested in following your example and doing something similar, are there any particular challenges or pitfalls that you encountered that people should look out for?
Yeah, thanks for the question. I have to admit that I haven't been involved from the earlier stages. So I would recommend definitely reaching out to us if anyone was going to wanting to model off metalabR. Yeah, we're definitely happy to collaborate, and we have, yeah, if you check out our GitHub and everything, that should be a way to find out, or if you've got specific questions, definitely ask us.
Great, thank you so much. That's a really lovely offer. So we have a few minutes left. Does anybody have any other comments or questions, or was it worth doing this? It sounds quite complicated combining bibliometrics and systematic mapping. I know how painful systematic mapping is, but then the idea of within the same project going back and trying to do something that I really have no idea about just sounds awful. But it's obviously really, really useful. And I had so many questions about, like, you know, we use these 350 search terms, vegetated strips, and some of them weren't so useful. We found some extra ones. Why are people referring to the terms they are? Are they siloed? Are they not connected? We had all these questions, but the systematic map didn't allow them to be asked because it was implicitly asking about citations and networks and not answering our users' question, which was, do buffer strips work and what are the adverse effects? Does anybody have any reflections about, I think there has been a discussion about the different users, but yeah, is it worth it was my initial question.
I mean, I would,
okay, go ahead, Sarah.
Well, I mean, I was just going to say, I don't know, you know, again, being in the very early stages of the project, it's hard to answer that question, but I mean, the evidence, you know, there's so much work that goes into the systematic mapping piece of this. And you have, you know, a really amazing data set with not just, you know, well-screened studies that meet a very specific, policy or practice relevant question, but also a lot of extracted data that you go through to, with each study. It's just like a really useful data set that you're missing this opportunity to even take that a step further. So I feel like, if we can sort of show and provide, open source tools that make this, more routine. I don't know. It seems like it's a great opportunity to learn so much more from this data. I don't know, Shinichi.
Yes. Yeah, adding to this, like tools which can like comprehensively do all those things are still limited. So there's a lot of opportunities there. And in the meta-analysis systematic reviews, this risk of bias is really, really big thing. And we have tests for the publication bias or time lag bias. That's really already social aspects, but the particular bias we cannot do in a meta-analysis or systematic reviews, actually citation bias. So citation bias, already actually, this field is like organism, and some papers are somehow, some people, some groups are a lot more cited than others, And actually bibliometric analysis, none of us really talked about it. I wanted to ask Tim and Dorian whether they thought about it. Citation bias is something like we can really map out current sort of state, how the influence, like how particular paper, group of authors are shaping this particular research field. And, is there anything we could do about it? Is that like a good influence? So it's actually the tool for meta research, meta science. And I agree with Sarah that maybe, listening to Tim and Dorian's talk, it's a lot, maybe this tools a bit too much. So you could certainly do systematic reviews separately, because systematic review is more like, getting at the evidence, researcher wants to know, practitioner wants to know. But the meta science is a little bit, it is for scientists, but it's very different, the question we are asking. And it's a really important, really, to accompany for the systematic reviews of meta-analysis.
That's a really great point. Thank you.
Yeah, one thing that we came up with over the course of analysis was bias in the form of industry bias. So in Canada, we had our bibliometric and systematic mapping really biased towards 3 or 4 major industries: forestry, peatland extraction, oil sands, and recovery in a particular city in Ontario. Now it's not really reflective of the ecological restoration practice in Canada, because a lot of that is in the gray literature, which wasn't captured by our 2 steps there, because it's so hard to find standardized gray literature or databases where we can search through them. So this bias towards those 2 or 3 or 4 industries was a product of big institutions partnering with industries, having the funding to partner with universities that produced academic literature. So a big struggle of ours is to sort of capture something that's not in academic literature, that isn't, a bunch of research groups citing each other quite a bit, producing these sort of thematic bubbles. And that's sort of where the other teams in our research group will hope to capture that. So they're doing interviews with practitioners, mostly non-academic, and taking case studies and sort of doing a deep dive into them, sort of trying to find these records of non-academic literature that document these restoration practices. So we're kind of going all in to try to reduce that bias as much as possible to get a complete idea of ecological restoration in Canada, something that really hasn't been done before.
Great, thank you so much. Just before we finish, we have one really, hopefully really quick question for Sarah, I think, from Camille Bellman. What are, in your view, the main network indicators you can use for these networks? So not thinking about visualization, but actually quantifying it, I think, or qualifying it rather than visualizing, which seems quite useful, which he suggests.
Yeah, I mean, I think there's a number of sort of standard like network level statistics and node level statistics. So for example, at the network level, you can look at network density or the number of components and look at how that changes over time. So network density can tell you something about how information is flowing across a network. The number of components might say something about how siloed or disconnected a network is. At the node level, you have measures like centrality, degree centrality, betweenness centrality, which can tell you something about the importance of particular actors or papers in a network. Betweenness centrality is an interesting measure that can say something about bridges. So for example, someone with high betweenness centrality would be potentially a real connector between otherwise disconnected communities. So I think there's a number of sort of standard statistics that provide more information than just a visual picture, but I don't know if Shinichi has additional thoughts on that. I'm still new myself to this kind of work, so.
Shinichi, do you have a 10-second answer?
I think Sarah covered it really well, I think. So no additional stuff, but yeah, it is pretty complicated. But you know, there's a lots of software to do network analysis, which can be like used as a, you know, the new variables fit into the statistical model. So yeah, so it's not that, you know, not as scary as it sounds.
Yeah, there's a lot of jargon when it comes to this kind of. So once you get a handle on that jargon,
you made up a new one as well.
Right? Yeah, right.
Brilliant. Thank you so much. We have to wrap up there. This was a really interesting session. Amazing talks. Thank you so much for the presentations and the panel discussion, Shinichi, to everyone as well for their talks. Just a reminder to the registrants to vote for their favorite talk. Join us again in just under 15 minutes for a session